further ado, I let Naomi prepare today.
All right. Hi, everyone. I'm Naomi Huntley. So I'm really excited to be hosting Dr. Sarah Clark today. So I'll just share a little bit about her background. So she attended the Perelman School of Medicine at the University of Pennsylvania with a concentration in microbiology, virology, and parasitology in the lab of Jeff Weiser. And then she did a postdoc in immunology with Laura Lenz at University of Colorado. And then now she is at the Department of Otolaryngology, I hope I said that right, at the University of Colorado in Denver, where she started her lab in 2019, just before COVID got going. And she's now there as an assistant professor, where she uses her dual training to investigate host-pathogen and microbiome interactions in the respiratory tract. And then I just wanted to highlight a couple awards that she's gotten. So she was featured as a new investigator award from Central Society for Clinical and Translational Research, Early Career Faculty Award from American Association for Immunologists, and Innovation, and she received the Innovation Award funding from the American Lung Association, I guess as part of the American Thoracic Society and NIH. And then outside of just research, she enjoys mentorship, where she's using that to increase retention of women in science. And then finally, she is I'm not sure how to say it, Botcher investigator with that foundation, which is a philanthropic organization whose mission is to invest in the potential of Colorado through scholarship, biomedical research, and capital grants. And today she'll give her talk titled "The Species-Specific Impact of Airway Prevotella on Infection Resilience in the Lung." So let's all welcome Dr. Clark.
Thank you so much for that introduction and also for the invitation to come present to you all. I'm very excited to visit and speak to some great scientists here, and it's been fun learning about the graduate program as well. So thank you for the invitation to come. So my talk title is a little bit different, but I promise I will talk about that title as well. But yeah, I'm interested in the airway microbiome and how that impacts host pathogen interactions. And so today I'm going to talk about a specific project focused on that. And see if I can advance this slide. Okay, so my research specifically, I'm really interested in respiratory tract infections, which with the pandemic, I don't have to convince you is an important problem to global health. But even before the pandemic, respiratory tract infections were a leading infectious cause of death, and especially impactful in people over 65 and under the age of 5 years old. But in the airway, I'm not looking at viruses. I'm really interested in the bacteria and bacterial pathogens of the airway. And I've highlighted some major players here, including Streptococcus pneumoniae, which is the major pathogen that I investigate. And what I find interesting about these pathogens in the airway is they exist kind of in this in-between space between commensal and pathogens. So they're opportunistic pathogens. So sometimes they're in our airways asymptomatically and not causing any disease, whereas other times, they're causing significant morbidity and mortality. And so a central question for my lab is trying to understand what host factors are tipping the scales here in terms of governing resistance versus vulnerability to these bacterial infections. Okay, so just to introduce my pathogen a little bit, Streptococcus pneumoniae, also known as the pneumococcus, a very clinically relevant bacterial pathogen. So outside of viruses, this is the most common cause of pneumonia. And we do have vaccines against this bacteria, but they're serotype specific. And so there's now over 100 serotypes of pneumo in circulation, and the vaccines just don't have the coverage to protect against all of these different serotypes. And another thing about the pneumococcal vaccines is that they're really good at protecting against invasive disease, but they're not as good at protecting against mucosal infections like infections in the lung. And so I think there's a real need for better prevention and treatments for strep pneumo infection. So in terms of thinking about what are the factors that control this balance and tip the scales between resilience and vulnerability to infection, the microbiome plays a huge role. And I promise that the microbiome in the gut is also important, but for my lab, I'm really interested in the microbiome that's present in the airway in the same place as the pathogen is infecting. And so to introduce this a little bit, the airway microbiome is this really diverse community. It's dynamic, it changes over your lifespan. And what I want to highlight here with this figure is the microbial density. So what you can see is that there's much higher burdens of bacteria in the upper airway compared to the lungs. So this is really what we think of in terms of the home for a lot of these bacteria, where they're very active, dynamic communities, and they're colonizing the upper airway, and you get aspiration into the lung periodically. And what's interesting about the airway microbiome is that outside of animal models demonstrating that it's important for resistance against infection, you can also look at these really nice longitudinal studies that have been done looking at sampling of the upper airway over time. And you can see that the composition of this community is predictive of infection risk. So that really supports this idea that this community is important and that changes in this community can impact resilience against infection. And so for this project, the bacteria I'm interested in are in the oral cavity and oropharynx, which some recent studies have highlighted is really the source of bacteria that are getting into the lungs, again, through this aspiration. So you're aspirating these bacteria constantly into the lungs, and it's seeding the lung microbiome. But when I think of that, I think of potentially an important immune priming signal. So how is this constant exposure to bacteria in our lungs tuning our immune response? And so why do I care about the immune response? Because it's really important for disease. And so when we think about these opportunistic pathogens like strep pneumo, it's colonizing very asymptomatically in the upper airway, so inducing a very minimal immune response up here. And when you get invasion to other host sites is when you get disease, and that you can think of it as this imbalance between our immune response trying to fight off the infection and bacterial replication. So after you get into a place like the lungs, you have disease symptoms due to your immune response to that disease. So this balance is really important. Okay, and so I wanted to highlight a couple steps. This is very oversimplified, but what happens once we get aspiration of a pathogen like strep pneumo into the lung? How do we successfully clear this pathogen? After aspiration, the first thing the bacteria sees and comes in contact with are the resident cells in the airway. So an important player here are alveolar macrophages and of course the lung epithelial cells. And one of the things they do is send signals, including chemokines, which recruit new cells into the lung. And so one of the first recruited populations are neutrophils, which are responding to the infection. You get a second wave of recruitment of inflammatory monocytes. And the reason that I'm really interested in these cell types is because they can directly contribute to killing of these bacteria. So they're taking up the bacteria, they're killing them actively. What's interesting though about these myeloid cell populations is that they can only control infection up to a certain point. After a certain point, these responses are overwhelmed. And so if it reaches that point, you need then this next step of development of a specific immune response, including specific antibodies to the bacteria to get clearance, because these cells have just been overwhelmed in terms of their ability to contribute to clearance. And so the reason I highlight this whole pathway is that successful pathogens like strep pneumo have developed a lot of mechanisms by which to avoid or evade or modify these signaling pathways and these responses so that they survive better in the lung and prolong and exacerbate these infections. And so what I think is important in then is before the bacteria are able to modulate these immune responses, the kind of readiness state or baseline priming of these immune cells in the lung is really critical in terms of the ability to rapidly respond to the infection before the pathogen is able to modify the killing activity and overwhelm these early immune responses. So I think a lot about the kind of state of readiness in the lung before infection. And so along those lines, in terms of what's governing that state of readiness, I'm interested in the question of how does the airway microbiome potentially serve as an important immune regulatory signature in the lung? And then if so, which bacteria are important and how are they contributing to protection? So the hypothesis for this project area has really been that bacteria which are abundant in the lung may influence the innate immune response to pathogen challenge. So we're just starting out with that very general hypothesis and trying to dig in to some of the mechanisms that might be important. So for the story today that I want to share, we focused on Prevotella as our kind of member of the airway microbiome that we think might be important for this effect. And so Prevotella are gram-negative anaerobic bacteria. And the reason I'm really interested in Prevotella is they're extremely abundant in the lung and the healthy human lung. And so if you look at, this is just one paper, but there's several papers, sequencing the airway microbiome and finding a very high abundance of Prevotella. And there's particularly abundant in the oral cavity, as well as in bronchoalveolar lavage fluid. And so it will vary a little bit depending study to study, but Prevotella are typically one of the top 3 most common and abundant bacteria that you find in the healthy lung. And the reason I'm interested in it is because this abundance negatively correlates with the pathogens I have of interest in my lab, Streptococcus pneumoniae. And also people with more Prevotella in their lungs are not the people who have pneumonia. So there's this negative correlation, which I find interesting. And there was one study in particular which highlighted a species of Prevotella, which is called melanogenica, which showed that this was the most discriminative bacterial signature, which separated people out who were healthy versus those who had pneumococcal lung infection, so infection with this pathogen. So I decided that Prevotella are interesting to look at, and we'd start out looking with this one species here as a representative member to start. And so I'll just abbreviate that here as PMEL. So we know a little bit about how Prevotella activate an immune response, and they're not completely quiet in terms of how the immune system recognizes these bacteria. So we know that they are inducing some immune responses. So in humans, we know that people who have more Prevotella in the lung have this subclinical inflammatory phenotype, which includes more activated neutrophils in their lungs. And we also know, if we work with epithelial cell lines, that Prevotella are inducing responses, including activation of the toll-like receptor 2. But this level of activation is not as robust as what's happening in response to pathogens. So they're inducing a little bit of signal, but not enough to cause an infection, and not as much as comparison with pathogens. And what's nice to see about the humans studies for someone who works primarily in mouse models is that some of these same pathways and steps might be important in mice when you get exposed to these same bacteria. So the Prevotella of interest that I have has already been shown to activate an immune response in the lungs of mice, and it's less inflammation than pathogens, and it's TLR2 associated. And I just wanted to highlight here, though, we don't think Prevotella might always be even beneficial or have no effect on disease, because there is an example in a mouse study where in a lung fibrosis model where there's already damaging levels of inflammation, exposure to this Prevotella actually worsens lung injury. And so the Prevotella are definitely not quiet members of our lung microbiome. They do activate some immune response, and we were curious how that immune response might be impacting susceptibility to bacterial infection. So here's a very simplified version of this hypothesis. We think that aspiration of Prevotella into the lungs, so a lung that has more exposure to this bacteria, might have a better chance at resilience against pathogen infection compared to lungs without as much Prevotella exposure. So to look at this mechanistically, we wanted to use a mouse model to see if some of these observations would hold true and if we can look at the mechanism of this effect. And so we developed this model looking at very acute infection responses. So we're using black 6 mice, and we just expose them directly into the lung, so intratracheally to Prevotella, and we've used live and heat-killed Prevotella. And then 1 day later, we're infecting with our pathogen, Strep pneumo, and then looking at often 24 hours post-infection. There's most of the data, which I'll be sharing today. So a very acute infection model, looking at those very early steps after exposure to the pathogen in the lung. And so what we found is that this is looking at burdens of Strep pneumo in the lung. And so each dot is the burden of the pathogen in the mouse lung. And so here's mice that were just pre-exposed to PBS, versus all the purple dots are mice which were pre-exposed to the Prevotella. And what I'm showing here, this is an equal dose of Prevotella to what we infected with in terms of the pathogen. And then we titrated down this dose. And so what we saw is a very striking and robust effect of the Prevotella. This is live Prevotella in this graph, where this is a limit of detection. We actually weren't able to detect any mice infected with strep pneumo who were pre-exposed to Prevotella in this experiment. And even titrating down the dose, we still saw significant protection. And so we also tried this with a higher infectious dose and looked at survival of the mice. In this case, we're using heat-killed Prevotella, and we see that there was a significant improvement in survival after this infection, again with pre-exposure to Prevotella. So this is just setting up a model where we think is replicating potentially something that's happening in people as well, but now narrowing it down to this one bacteria and one pathogen, so we can look at the mechanism of how this might be occurring. What was really promising and got me more excited about this project is that while we were doing that, another group published a very similar finding, which is always nice to see that your data can be done in a completely different lab with a completely different group and see the same thing. So what they were doing was this mixture of live oral commensals, they called MOCs, which included the Prevotella that I'm working with. And they found when they pre-exposed, so this model is just like the graph I showed you before, but with this 3 bacterial mixture, where they were pre-exposed with Prevotella, they saw reduced infection of strep pneumo in the lung. And what's interesting about their data is that the protective effect lasted up to 14 days later. And I don't know if this is due to the Prevotella or some of these other species, but it was a long-lasting effect in this model system. And then similar to us, it also increased survival. So it seems like there's a robust effect here, which is repeatable. And we wanted to focus first on these initial responses. What's causing the difference here in this early clearance phenotype? And so I've divided the talk up into kind of 4 different sections. And so, as I mentioned, I first wanted to look at the early immune response, and I showed you one of the earliest steps that happens are the recruitment of these myeloid cells, which help to kill the pathogen. So I wanted to look at that first. And so when we started playing around with this model, I'm showing here heat-killed Prevotella, again, see a protective effect. We wondered if just any heat-killed bacteria would do the same thing. And so we've tried a number of other bacteria which don't have the same protective effect, including just heat-killed E. coli. Another thing we tried is just E. coli LPS. So the reason we did this is because LPS is well studied as a model of acute lung injuries. So we know a lot about the type of inflammation that this exposure will cause in the lung. And I thought it was interesting that we don't see a protective effect here, because it made me think maybe we can use this as a comparative tool. So how is LPS from E. coli activating the immune system versus how Prevotella is? And maybe that will give us some clues in terms of what's relevant to this protective mechanism. Yeah, of course. So no, it's not. So it's like gut. And so I'm just here using it E. coli, and I'll talk about this a little bit later. Yeah, the question for anyone who didn't hear is E. coli like relevant to the lung in terms of a normal exposure? No. If you get really sick, you'll get some E. coli into the lung because of likely barrier leakage is happening. Otherwise, no.
Perfect negative.
A negative control, and also the LPS, I'll get into this later, is the LPS of E. coli is a little bit different than commensal bacterial LPS too. So it's even different than commensals, I think, in terms of an exposure. Exactly. Okay, so to simplify things and just first look at the immune response to the Prevotella versus the LPS, we're now not doing a co-infection here. So we're just looking at Prevotella or LPS, and then 1 day later. So this is the time point at which normally I'm infecting the mice with the pathogen. So first thing we looked at, the recruitment of these different myeloid cell populations. I'm showing neutrophils here. This is by flow cytometry. So looking at the immune populations in the lung, I've gated on neutrophils here. And neutrophils, in addition to other cell populations in the lung, look very similar between Prevotella and E. coli. So we got similar levels of recruitment of several cell types. But what was interesting is that we saw a specific effect on TNF-alpha expression in neutrophils for the Prevotella exposed mice. So TNF-alpha is a pro-inflammatory cytokine. You can think of it as this kind of generalized activation marker for myeloid cells. And we saw that the Prevotella activated TNF-alpha response, whereas E. coli didn't really activate this response in the neutrophils. So that got us interested in neutrophils and the inflammation associated with TNF-alpha. So one thing that we've done to see whether or not these are important is just do an antibody depletion model. And so we're going back to co-infection here, where we see the Prevotella protective effect. And here in this graph, I'm using neutrophil depletion antibody versus a TNF alpha depletion antibody. And you can see in both cases, when I add in the depletion antibody, even though I still have Prevotella has been exposed, the protective effect is lost. So without neutrophils, you don't get protection. And without TNF alpha, you don't get protection, highlighting that we think these are relevant to the mechanism here. So just to describe a little bit about neutrophils, because now we had some evidence that looking at inflammation and looking at neutrophils might be relevant to how Prevotella are increasing early clearance. Neutrophils can kill bacteria through several different mechanisms. I'm showing some of them here. As I mentioned earlier, they're really important for early bacterial pathogen clearance. For strep pneumo specifically, the two responses which seem to be critical for neutrophil killing of this pathogen are the production of reactive oxygen species and the activity of these serine proteases. One of them is elastase, which I'll show some data with. So that's some of the mechanisms. Other pathogens can be caused by netosis, but it's not as effective against this pathogen specifically. So neutrophils can directly kill this bacteria. We think they're important due to the antibody depletion experiments. They seem to be producing TNF alpha, but we wanted to know, are they better at killing this pathogen directly? And that could be a mechanism by which they're important. So to ask this question, we tweaked our model slightly again, where this time we're looking at exposure to the Prevotella or LPS, and then we're purifying neutrophils from the lungs of the mice 24 hours post-exposure. So again, this is the time point at which normally I would be challenging the mice with the pathogen. And so what we're looking at is neutrophil killing of strep pneumo. So there's a well-established killing assay where you incubate strep pneumo with the neutrophils and then just look at how many bacteria are able to be killed. And what we found is that the neutrophils from the lungs of the Prevotella exposed mice were just these super killers. We saw enhanced killing by neutrophils from the Prevotella exposed mice compared to the LPS exposed mice. And I'm not showing the data here, but a naive mouse doesn't have as many neutrophils in the lung, but if you pool a bunch together, the killing looks more like this rather than this. So it's not that this is suppressive, it's rather that there's an enhancement with the Prevotella around. We looked at the mechanism of this effect, and I'm not showing all the data here, but we looked at the reactive oxygen species production was very similar between these groups. But what we did see was an effect on the serine protease activity. So here you see increased elastase in the Prevotella exposed mice compared to the LPS exposed mice. And when we did a protease inhibitor, we saw this effect go down. So we think that there's a direct impact of Prevotella exposure in the lung environment to enhance neutrophil killing. So they're just better primed at killing strep pneumo, and it's likely through this serine protease activity. So our model initially here is that exposure to Prevotella in the lung is enhancing an innate immune response, including through recruitment of neutrophils, which are more activated. They're producing TNF alpha, but also they're just better at killing strep pneumo. So the next question we wanted to know are what signals on the Prevotella are interacting with what receptors on the neutrophils to activate this protective response. And so I said I was going to bring up LPS again. I have to go into a little bit about what signals, why we chose to look at the signals we did. So Prevotella are gram-negative, but their LPS is under-acylated. So E. coli has an LPS like this, which is hexa-acylated and is very good at signaling through TLR4. Under-acylated LPS is not as good at signaling through TLR4, and in fact, in some cases can be inhibitory to TLR4 signaling. So bacteria with under-acylated LPS actually signal through TLR2 much more predominantly. And this matches with the data I said previous groups have established that Prevotella signal through TLR2. And the major bacterial ligand of TLR2 are lipoproteins. So we wanted to look at lipoproteins and their signaling through TLR2. Okay, so again, we use a slight variation of our model. This time we're using naive mice and just taking neutrophils purified from the bone marrow and exposing them to Prevotella directly. And we're looking at neutrophil activation here by the secretion of TNF alpha into the supernate of these cultures. So here I was able to isolate neutrophils from wild type versus TLR2 deficient mice. So when we look at their activation by TNF alpha, we see that exposure to the Prevotella induces this nice TNF alpha response, which is completely gone in a TLR2 deficient mouse. If we purify LPS from Prevotella, we can see some TNF alpha production, but it's not TLR2 specific as you would predict. But if we purify lipoproteins from the Prevotella, we see again this nice TLR2 specific induction. And so the lipoproteins seem to be sufficient to activate a TLR2 dependent response in this very simplified in vitro system. Another thing that we've done is treat our Prevotella with a lipoprotein lipase. So that digests some of the lipoproteins enough so that they can't signal anymore through TLR2. And what we found is that this lipase treated Prevotella was no longer activating this response. So we see in vitro some relevance for this lipoprotein signaling through TLR2 axis, but we wanted to know if that was relevant to the protection model we see with our co-infection in vivo. So again, we're turning back to the mice and looking at Prevotella exposure followed by pneumococcal infection. And so what we found is that in contrast to our untreated Prevotella, if we use our lipase treated Prevotella, we saw it was no longer protective, suggesting that these lipoproteins are an important mechanism for the protective effect. What was interesting, however, is that we couldn't just use purified lipoproteins from Prevotella to get protection. And another thing we've tried is different doses of this TLR2 agonist to just activate TLR2, and that also is not protective alone. So it's not enough to have this TLR2 lipoprotein signal. You need additional signals, but lipoproteins are important, just not sufficient for protection, we think. And so with TLR2, the way we looked at relevance for this signal was, again, looking at our wild type versus TLR2 deficient mice. You can see in the TLR2 deficient mice that there's just no longer a protective effect at all of Prevotella. And if we look at the neutrophils in the lungs of these mice, we see in contrast to the wild type mice where we saw that elevated TNF alpha, that activation signal, that also is lost in the TLR2 deficient mice, suggesting that this TLR2 mechanism is really required for protection. Another thing we did is go back to our model where we purify neutrophils from the lungs after Prevotella exposure, and then look at killing of strep pneumo. And so you can see compared to the wild type neutrophils from wild type mice, which were exposed to Prevotella, there's nice killing. The neutrophils from the TLR2 deficient mice were not as good at killing, and they also didn't have very much serine protease activity. And so our working model, which I think is oversimplified at this point, but a picture that's starting to emerge is that Prevotella lipoproteins are important for this protective effect. We know that they can signal through TLR2 on neutrophils, probably are signaling on other cells as well. And that one consequence of this signal is the production of TNF alpha by neutrophils. So we've seen that in the lungs of the mice, as well as the secretion of this in vitro. And then also this enhanced serine protease activity, which seems to be correlated with the ability of the neutrophils to kill strep pneumo directly. Now, what's interesting, I didn't really highlight with the data when I was showing it, but we think that these two responses might be somewhat require a little bit different signaling, because this neutrophil TNF alpha response we got when we just looked at our purified neutrophils from naive mice and stimulated them directly with Prevotella. But this increased killing phenotype, we always were using neutrophils purified from the lungs of Prevotella exposed mice. And we don't see enhanced killing when we just expose naive neutrophils to Prevotella. So we're missing some intermediate cell, which is needed to enhance this killing effect. It's not just the neutrophils seeing Prevotella, it's probably this plus additional signals. So next I want to turn to looking at the restraint of this inflammation. And the reason, so I've been talking a lot about activation of neutrophils, activation of TNF alpha, inflammation. All of those things are great for killing pathogen, but they're very damaging to the lung. And I think in addition to worrying about bacterial clearance, you also have to worry about inflammation in the lung and restraint of that inflammation, because the lung is especially sensitive to inflammatory damage. So even after you clear the pathogen, you can get a lot of morbidity and mortality due to the damage that's caused in the lung due to inflammation. And there's this kind of established Goldilocks principle for TNF alpha during bacterial pneumonia, where you need just the right amount. because too little of this response and you can't clear the bacteria from the lung, too much of this response and you die from tissue damage to the lung. And so I was wondering about whether or not this balance was important for the Prevotella effect that we're seeing. And specifically, I was interested in looking at this cytokine IL-10, which is a major anti-inflammatory regulator. And previously, I did a lot of work looking at the consequences of IL-10 in different infection models. And so what we know is that increased IL-10 can either have protective or detrimental effects, and the kinetics matter a lot in terms of the outcome. So some pathogens, including strep pneumo and also listeria, seem to activate a delayed IL-10 response, which promotes their survival. Whereas in this lethal cerebral malaria model, we found that IL-10 was extremely protective. So I think the balance of IL-10 and the kinetics of IL-10 are really important. So even though we know that IL-10 at a delayed time point is bad for clearance of strep pneumo, I wondered if earlier IL-10 might actually be protective in this Prevotella exposure model that we were looking at. And the reason I was interested in this is because it's been shown previously that this Prevotella strain we're working with is able to activate IL-10 production from immune cells in the mouse lung and also human myeloid cells. So we wanted to look at IL-10 and its potential contribution in this model. So I wanted to summarize a lot of our data into a schematic to just show you some of our findings so far with this. When we look at Prevotella exposure, that I showed you the neutrophils are making TNF-alpha. This is associated with a systemic TNF-alpha response, and we also see systemic IL-10. And this is the time point at which the mice are being challenged with strep pneumo. Now, after infection, 1 day later, when we're seeing clearance of the pathogen, we actually already see the suppression of a lot of the immune stimulation responses are going down. So we see a reduction in the systemic levels of IL-10, but we still see elevated, we see a reduction in the systemic levels of TNF-alpha, that pro-inflammatory signal, whereas we still see elevated IL-10. So that was interesting, particularly this reduced systemic TNF-alpha, which was different from mice infected with strep pneumo alone. So I wanted to know, what is the impact of this IL-10 in terms of the Prevotella model? And it was an easy question to test because we've got IL-10 deficient mice. And so we just looked at the Prevotella co-infection model. Here's in wild type mice, just showing the same phenotype. And in IL-10 deficient mice, it actually was not protective at all. So that was really interesting. And we were confused as to why this would occur. IL-10 is an immune suppressive signal. It's not supposed to be protective. But when we looked in the lungs of these mice, what we found is an overwhelming inflammatory response. And I'm just showing neutrophils here, but pretty much all the myeloid cells that I looked at had a similar phenotype where there's just this robust TNF-alpha response in the absence of IL-10. So you really need IL-10 as like a brakes on the system in terms of the amount of inflammation. Otherwise, this inflammation, which is induced by Prevotella, is no longer protective. So our working model to describe these findings so far is that we think there's an environment in which you have Prevotella immune priming compared to in the absence of immune priming that's more protective against challenge with strep pneumo. So what we think are the important steps is that after exposure to Prevotella, you get recruitment of neutrophils. These neutrophils seem to be activated through a mechanism including lipoproteins interacting with TLR2. And one of the consequences of this, in addition to inflammation associated with TNF-alpha, is direct killing of the pathogen, but that all of this needs to be controlled by an IL-10 response. And so I've been showing you all data with one strain, one isolate of Prevotella. And one of the questions we were interested, after we've established a working model of at least knowing some of the important players, I wanted to know, is this a general feature of Prevotella? Are all strains of Prevotella protective? And what's interesting about Prevotella is there's a lot of genetic variability even within species of Prevotella. And then in addition to that, there's kind of a spectrum of Prevotella in terms of commensalism versus disease. And so you can say there's a spectrum for pretty much any bacteria. Some are much more on the commensal side and some are much more on the pathogen side. And I pulled this figure from a really nice review, which is summarizing this kind of spectrum with Prevotella different species. And what they did in this review is in the gray bars, the thickness of the bar correlates with how many reports are associated with different infections for different species of Prevotella. And I just want to show this because I want to highlight there's some species of Prevotella which are more highly associated with infections compared to others. So no Prevotella is important enough to be considered this major human pathogen, but there are some Prevotella which are definitely more pathogenic than others, I will say. And also there's difference in virulence factor expression for some of these species versus other species. So specifically intermedia and nigrescens are associated with a lot of oral infections compared to Prevotella like P-mel that I've been working with. Another interesting thing in terms of looking at different species is that there has been some work on intermedia because of its importance in periodontal disease. And others have tried a little bit of a different model, but still looking at a peritoneal infection model. And they saw that intermedia did not increase neutrophil phagocytosis. So I was interested, because I see an enhancement of neutrophil activity with P-mel. Does P intermedia have a different response potentially? And also another group showed that P intermedia increases expression of this receptor, which can be used by strep pneumo to adhere better to the airway epithelium. I'm just showing their data here where they're looking at expression of this receptor going up in response to P intermedia and combined with strep pneumo. And then they're looking here at an adherence of the pathogen to the airway epithelium. So this potentially would be detrimental, not protective. And so I wanted to compare intermedia and other Prevotella species to the isolate I've been working with. So we now have a collection of Prevotella strains. We're still working our way through these strains. So some of these data are still unpublished and we're still working our way through the collection. But what we've been doing is exposing mice to live Prevotella and then infecting as one of our model systems. And so what we found is that the Prevotella seem to be associated into two different groups. And so in one group here in the gray circles are all Prevotella, which have a very similar phenotype to the original Prevotella strain I've been talking about. So this is a different isolate of P-mel. And then these are three different species of Prevotella, which all behave very similarly. We see enhanced production, and we've actually expanded this number of protective Prevotella here as well into additional numbers. But in addition to these protective Prevotella, we saw these other Prevotella which were not protective. And what immediately stands out is that these are the two Prevotella I highlighted that are more highly associated with disease. So intermedia and nigrescens, associated again with more oral infections, they were not protective compared to P-mel. And so we said, okay, that's our in vivo phenotype. How are these different Prevotella interacting with neutrophils? So we went back to our mouse where we're taking naive mice, purifying neutrophils from the bone marrow and exposing to heat kill Prevotella. And what was really interesting is that the protective phenotype correlated with how these different Prevotella are interacting with neutrophils. So the protective Prevotella, like this different isolate of P-mel, activate the neutrophil secretion of TNF alpha in a very TLR2 dependent manner. So that's true for all these protective ones. In contrast, these non-protective Prevotella had TLR2 independent activation. So they're interacting with neutrophils in a different manner than these protective ones. We saw a similar phenotype when we looked at the enhanced killing effect of Prevotella. So again, we're purifying neutrophils from the lungs after Prevotella exposure. And in contrast to P-mel, which enhances killing, neither intermedia nor nigrescens had that same enhanced killing effect. So in going back to our model here, we think our conclusions are a little bit different about Prevotella priming versus not priming, because we think that the species of Prevotella might be really important for this. So we've isolated a few different protective Prevotella, which probably act very similarly in terms of, at least in our initial observations with neutrophils. And then we've isolated some non-protective Prevotella. We're not sure at which stage of this pathway, whether it's interacting with signals upstream before the neutrophils, as well as these interactions with neutrophils, which seem to differ from the protective Prevotella species.
but it is interesting that phenotypically the difference between the protective and non-protective do correlate with how they're interacting with neutrophils and TLR2. So it just again highlights the importance of that signaling pathway for this protective mechanism. So I wanted to leave, I have just a couple more sections of more speculative and preliminary data that I wanted to share just in terms of our thinking of outside of pneumococcal infection and Prevotella exposure. So we talked about different species of Prevotella. I'm also interested in how much this applies to other infections, not just pneumococcal infections. And one of the reasons we became interested in this is that Prevotella, in addition to being very common in the healthy lung, are also common in the cystic fibrosis lung. And so in the cystic fibrosis airway, you have much higher microbial density, including more anaerobic bacteria such as Prevotella. But there's still a lot of debate in terms of if they're helpful or harmful in this disease context. There have been, however, some reports that associate Prevotella dominance with higher lung function compared to individuals with CF who do not have a dominant Prevotella profile. And then also the people who have more Prevotella tend to have it's before they get infections with these really bad pathogens like Staph aureus and Pseudomonas aeruginosa. Those patients don't have Prevotella in their lungs. So we were curious if this protective mechanism might apply to other pathogens, including those relevant to cystic fibrosis, as an initial way to kind of address how Prevotella might be acting in the context of CF. So this is very preliminary, but we have done some experiments using now Staph aureus in the same co-infection setting. So just looking at Prevotella exposure and then challenge with staph. And so what we saw was the effect was not as striking as with pneumo, but there was some impact. If we look at heat-killed Prevotella, we see that there's now a reduction in Staph aureus infection in the lung. Certainly not this total clearance effect, but definitely a reduction. This is several experiments now, which we've done, replicating these findings. And then we also looked at neutrophils from the lungs of mice after Prevotella. So the same kind of setup I had for strep pneumo, and looked at killing of staph. And what was promising so far is that we do also see this enhanced killing mechanism. So potentially that difference in clearance might be related again to this enhanced neutrophil killing phenotype, including neutrophils' ability to kill staph. And so this is very much in progress work. We're screening now more Prevotella isolates sent to us from patients with cystic fibrosis from Paul Planet at CHOP. And we're interested in how different species of Prevotella and even different isolates from the same species might impact these immune responses that we're seeing. It could provide some protection, at least against staph, which is a very relevant CF pathogen. And then I wanted to end by not ignoring the gut, because I know there's a lot of gut microbiome people here. And so I just wanted to mention, I'm thinking also about how Prevotella might be acting in different mucosal sites. So not just the lung, we also have Prevotella in the gut. There seems to be a different enrichment of Prevotella species in the lung versus the gut. So like P. copri and Stercoria are the more dominant Prevotella in the gut. And P. copri is famous as a potentially immune modulatory species in the gut, but we're arguing over whether or not it's good or bad, similar to some of the Prevotella literature I talked about. But it was interesting, I just wanted to highlight a body of work from a group looking at oral exposure to Prevotella histicola. So this is an inactivated treatment with histicola, and it's being developed as this broadly anti-inflammatory therapeutic. It seems to induce a T regulatory response and be suppressive of type 1 immunity. And it has this interesting effect of helping to reduce symptoms from a variety of diseases, including the experimental EAE model for multiple sclerosis. And what's interesting about this is that it's shown efficacy in a phase 2 trial for psoriasis in people. So I thought this was interesting to compare what's known about this Prevotella interaction in the gut with this oral therapeutic. And in looking through this literature, they actually directly compared the same female that I'm working with this histicola treatment. And so what this is just clinical scores from this mouse model of EAE for, it's a mouse model of multiple sclerosis. And here's the histocola, which is more protective than P-mel, although P-mel had this intermediate effect. So in the gut, this bacteria won over this one in terms of protection in this disease setting. What was interesting is that they found that this was a TLR2 dependent effect. So TLR2 was also important for histocola. And in looking through their data, their P-mel oral inoculation, which was, every other day for several days, that induced a systemic IL-10 response, which I said we saw with our lung exposure. And also they saw reduced TNF alpha, which again, we saw in the lung exposure. And these signals were present by day 14, so an extended period of time. So it's been really interesting to compare this data with our data in the lung, because I think some of these signals that we see for exposure to the lung might be also activated in the gut with this oral exposure. Okay, so just to highlight some of the conclusions that we have, some more preliminary than others, but we think that the effect of this Prevotella in terms of the immune response and how it might generate protection is through activation of neutrophils and a signaling access, which includes lipoproteins on Prevotella and TLR2 on the host. But I think that there's much more to be discovered here. This is required, but not sufficient for this whole pathway to happen. So I think there's other things going on in terms of other factors on the Prevotella which are relevant. Another, I think, interesting finding from this is that deregulation of this protective response is just as important as inducing the protective inflammation. So regulation by IL-10 is really critical for controlling the amount of Prevotella inflammation. And also protection with this Prevotella seems to be species specific. And this specificity correlates with how they're interacting with neutrophils. So I think there's a lot of exciting future directions that we'd like to work in this project. I talked a lot about neutrophils and kind of ignored all the other steps that I went through at the very beginning. So we're interested beyond neutrophils, like how Prevotella is interacting with those resident cells in the lung. So who's calling the neutrophils into the lung in the first place? And then what's the long-term impact? Like how long are these Prevotella exposures protective? And I'm also interested in looking at mixed settings, especially now that we know that different species have a different effect. So what happens in an actual person when you have a complex mixture of bacteria, including, probably several species of Prevotella, which might be present. And then I just want to leave you with, I think it's also important to consider the different contexts in which Prevotella might be harmful versus beneficial, because it is inducing a little bit of inflammation, which might be too much for some cases in terms of a chronic lung disease and inflammatory disorders. I'm just not sure what the outcome will be in terms of it does generate some immune regulatory response, but it's also inducing some neutrophils and activation of these cells. So I think it will be really critical to tease apart the impact of Prevotella on different disease settings as we think about developing kind of airway immunobiotics or therapeutics similar to those in the gut. And so with that, most importantly, I want to thank my lab. These are the contributors mainly to the Prevotella project, and continue this project through COVID after just starting my lab. So these are the real stars here of the show. I want to thank our collaborators, all of whom have sent us strains critical for this project, as well as my funding. And then I'll be happy to take any questions. Thank you.
Thank you, Sarah, for that beautiful talk. And yeah, we're gonna start with questions. We like to start with you. So I think there's a question right there. Yeah.
Yeah, that's a great question. She's asking, I don't know if everyone heard about the type 6 secretion system and potentially direct bacterial interactions. Yeah, I'm interested. We did try our, live P-mel with strep pneumo, and there was no, strep pneumo did not care about, there was no direct competition between P-mel and strep pneumo. Now, the answer might change in terms of Prevotella competing against each other or other members of the microbiome. That certainly could play a role. I'm not sure for the lung example, for example, we're aspirating these things into the lung. There is a little bit of enrichment for certain species over others, and that might contribute in terms of them killing off some things. But yeah, that's something that I'm really interested in terms of like only some of the Prevotella have the type 6 secretion system. And so what relevance that has to these complex mixtures might become really important. Yes.
Thanks for excellent talk. Research. So I had a question that you found some of the species, and some of them did not. And you said that most of the species occurred in lungs through the aspiration from oral microbiome. So are all of these Prevotella species actually like the natural habitats of the lung, or some don't?
Yeah, very good question. So another reason we picked P-mel is there's not a ton of microbiome studies which go to the species level. But of the ones that do, that is one of the most abundant Prevotella in like the healthy lung and also in the healthy oral cavity. And intermedia is very common as well. So of the ones that I've shown, all of them were ones that are found in the airway, including the lung. I'm limited somewhat to how many studies have a species level specificity to make the calls. But the ones that do, the P-mel that I showed and some of the other strains, among those P-mel is the most dominant. And then of the like non-protective ones, P intermedia is very common. Yeah, so I think that there's a real life scenario in which you would have different levels of P-mel and intermedia in different individuals. And so that's what I wanted to model next in the mice, is looking at like the ratios. Does it matter if you have like 50%, what's the outcome? Yeah. Yeah, so about distribution of Prevotella, from what I have asked lung microbiome people about this myself, it seems to be gradually less and less and less the further into the airway you go. For most of the orally aspirated bacteria, including Prevotella.
Thank you for your excellent presentation. You try to explain the protection from immunological perspective, but usually when there are two bacteria producing from immunological perspective, like this competition, and my second question is like protecting measure like media, using that kind of thing, how can you protect without completely risk measures?
Risk measures of like more severe
Like
disease.
relative risk, also like
Yeah, I'm using just a very general term in terms of clearance of the pathogen from the lung is the main thing I'm looking at. I'm not measuring things like lung function, like forced respiratory volume or something like that. I'm just looking, from a microbiologist standpoint, how much pathogen is still there versus not there.